All right, guys, let's talk about excretory system in protozoans. Excretory system in protozoans. And of course, as you can see, we have very nice diagrams drawn on the board to aid our understanding and comprehension of this. You just make sure you follow me every step of the way, and you'll be able to grab this quite easily. So the main organ of excretion in a protozoan like the amoeba, for example, or the paramecium, though our case study here is the amoeba, all right? The main organ of excretion is the contractile vacuole, All right. Before we get into today's conversation, I think it's really important that we understand osmosis as a term. Osmosis. So what is osmosis? Osmosis can simply be defined as the movement of solvent molecules from regions of low concentration to regions of high concentration. It's very easy. The movement of water molecules or solvent molecules from regions of low concentration to regions of high concentration via a semi-permeable membrane. I'll say that again in case you missed it the first two times. So osmosis can be defined as the movement of solvent molecules or water molecules from regions of low concentration to regions of high concentration through what we call a semi-permeable membrane. All right, the next thing we need to understand is we need to know the difference between solutes and solvents. Okay, a solute is the substance that dissolves in water, which is the solvent. Okay, the substance like a solid, for example, that dissolves in water can be called a what? Solute, while a solvent is the dissolving mixture like water, for example. I think these are really important terms. So osmosis involves the movement of water molecules up a concentration gradient. When I say up a concentration gradient, what I mean is from a region of low concentration. Okay, the water molecules are moving from a region of low concentration, all right, to a region of higher concentration. It's quite easy actually. All right, now that we've gotten that out of the way, let's shift our focus to the diagrams on the board. This is an amoeba cell, okay, and as you can see in this diagram, we have let's assume that this is an amoeba cell in a lake, a small body of water. All right, so the osmotic concentration or the osmotic pressure of that lake is low, as you can see written very nicely on the board. So low osmotic pressure. And then if you look inside the amoeba cell, you'll find that it has high osmotic pressure. Inside the amoeba cell, we have high osmotic pressure. I hope this is very clear. So based on osmosis now, please listen up. Based on osmosis, water will seek to move up a concentration gradient from a region of low concentration to a region of much higher concentration, okay? So due to that fact, you see that water in the lake will strive to enter into the amoeba cell. And of course, it's important you also know that the membrane of an amoeba cell and cell membranes generally are semi-permeable, meaning that they selectively allow substances to move through them depending on the size, the pore size, and all of that. You know, in another lecture, we'll look into that in more detail. So due to osmosis, the water molecules will try to move up a concentration gradient from, you know, the lake where there is low osmotic pressure into the cell where there is high osmotic pressure. This is very, very easy. Now, as that happens, a contractile vacuole is formed inside an amoeba cell. And contractile vacuoles could be, there are some that are spherical, and then there are others that are stellate or star-shaped. But that's by the way. So as water molecules enter into the amoeba cell, you find that the water molecules enter into the contractile vacuole through the membrane. You can see the lining around the contractile vacuole, right? So water molecules now enter into the contractile vacuole. Now let's shift our focus to the next diagram on the right. As water molecules enter into the contractile vacuole of the amoeba cell, what happens is that the water molecules continue to enter, and then the contractile vacuole continues to expand. It expands continuously, absorbing a lot of water until it reaches capacity. The moment it reaches capacity to the point where it can no longer, you know, take any water again, then we shift our discussion to the diagram on the right over here. Now, as water molecules enter, sufficient quantity of water has entered into the contractile vacuole of the amoeba cell. What happens is that the concentration gradient will flip. Now, what that means is that the osmotic pressure in the lake or in the water, the body of water surrounding the cell, becomes higher, and then the osmotic pressure inside the amoeba cell now drops. It becomes low. All right, you can see that, right? It's very easy. Now, when that happens, Based on our understanding of osmosis, water always likes to move up a concentration gradient from a region of low concentration to a region of high concentration. Please listen up. This is very, very easy. So now that the internal environment of the amoeba cell has a low osmotic pressure, the water will strive to, go out, to extrude, to be expelled out of the cell from the contractile vacuole, of course. Now, as you can see, the contractile vacuole then moves, it moves and then fuses with this cell membrane, the amoeba cell membrane over here. And then what happens next is that the water is expelled. As you can see very nicely here on the diagram, water out. Okay, the water is now expelled from a region of low osmotic pressure to a region of high osmotic pressure. And after that happens, you see that the membrane of the contractile vacuole then reunites with the cell membrane, and the process repeats itself. Now, this is a summary of how the contractile vacuole aids in the expulsion of waste products in protozoans. Now, water isn't the only waste product in an organism like protozoa, like the protozoans like the amoeba or paramecium or euglena. We also have a couple of waste products like ammonia. Okay, so as the excess water is being expelled, the ammonia, which could be toxic to the amoeba cell, will also be, since ammonia is quite soluble in water, the excess ammonia is also expelled alongside the water. Carbon dioxide is also a waste product in protozoans like this. It is expelled alongside the excess water, and these processes happen through the expulsion of ammonia, and carbon dioxide happen through the process of simple diffusion. Now let's get right into the next part of our teaching. All right, let's talk about the excretory systems in flatworms. Excretory systems in flatworms. We are done with that of the protozoans like amoeba, right? Now I want to look at the excretory systems in flatworms. The main excretory structure in a flatworm like planaria, like schistosoma, is the flame cell. The flame cell is the main excretory structure in flatworms. And before we get into the diagram on the board, it's important we do a brief overview of the flame cell or the excretory system in flatworms generally. Now, the excretory system in flatworms comprise of two longitudinal canals comprising of a network of ducts that lead to the exterior of the organism. It leads to the other structures or the other parts of the flatworm, forming a structure called the flame cell. The internal structure of the flame cell consists of an empty space basically called the cell lumen, and the cell lumen runs downwards, narrowing up and then forming the excretory ducts. Now, in the flame cell, there is a really important structure called the brush of cilia. The brush of cilia functions basically in sweeping down excretory materials. Okay, so the two longitudinal canals eventually link up dorsally. Dorsally means the back surface of the organism, and then linking to the excretory pore. Now that we're done with the overview of the excretory system of the flatworm, let's get right into excretory system in planaria specifically. You remember I promised to focus on planaria when discussing excretion in flatworms, right? Examples of flatworms include planaria, of course. Schistosoma is another one. Tapeworm is also a flatworm. Now, this is a diagram of a flame cell. Flame cells are basically bulb cavities. The shape of a bulb. Bulbs are something like this, right? So you can see this is quite similar to what we have here. So flame cells are bulb cavities that are funnel-like in shape as well. And as you can see, we have the cell lumen over here. The empty space you are seeing in the internal structure of the flame cell is called the cell lumen. As you can see, this dark staining part is called the brush of cilia. All right, and then as it narrows down from the cell lumen, it forms the excretory ducts. All right, now the major excretory materials in flatworms include carbon dioxide or carbon dioxide, as well as ammonia. Now, when liquid waste like containing carbon dioxide and ammonia diffuses into the cell lumen of the flame cell, what happens is that the brush of cilia sweeps this liquid waste until it enters into the excretory duct and then up until the excretory tubules. The excretory tubules then link up to the excretory pores at the dorsal surface of the organism until that waste is expelled out of the body of the flatworm, in this case planaria. Let's quickly review that before we conclude. Now, this is a flame cell, a bulb cavity which is funnel-like in shape. The internal environment, the empty space you are seeing in here is called the cell lumen. This is of course where the liquid waste, which could contain carbon dioxide or ammonia, enters in first. You can see the dark staining structure from the top over here is called the brush of cilia, and you know the function of the brush of cilia is to sweep down that liquid waste into the excretory ducts, down the excretory tubules, which link at the dorsal surface of the organism to the excretory pores, and then expelled out of the body of the flatworm. So this is the overview of the excretory system in a flatworm, of course in this case the planaria. Let's quickly move to the next part of our teaching. All right guys, let's talk about excretory system in earthworm. Now this is quite interesting, quite fascinating, so I'll say you listen up, you will really enjoy this one. Let's start with a brief overview of the excretory structures, or the excretory structure basically found in an earthworm. The main excretory organ of an earthworm is called the nephridium. All right, the nephridium. Let's talk about the nephridium for a second. The nephridium is the excretory, of course, the excretory organ of the earthworm. Each segment in the body of an earthworm, except the first three segments and the last, has a separate nephridium. Okay, and a nephridium basically, the couple of nephridiums you find in the body of an earthworm, they all have a similar plan. Their structure is the same. Okay, now a nephridium has 5 portions. Please listen up. The first portion is called the nephrostome. Okay, the second portion is called the narrow tube. The third portion is called the middle tube. The fourth portion is called the wide tube, and then finally we have the muscular tube. These are the 5 basic portions of a nephridium. Now let's get right into the mechanism of action of the nephridium. Let's begin with the nephrostome. The nephrostome basically is ciliated and funnel shaped, and the nephrostome is located in a separate body cavity called the coelom, and the other coiled structures are found in separate segments or body coelom as well. Now the body walls, the wall of the body cavity secretes what we call chloragogenous cells, which function primarily in the deamination. Deamination is spelled as DEAMINATION, in the deamination of amino acids into urea and other organic compounds. As these substances are deaminated, the beating action of the cilia then causes, as the substances are deaminated, we form urea and other organic substances. Those substances are then expunged into the coelomic fluid or the body cavity fluid. And then the beating action of the cilia of the nephrostome then draws in this coelomic fluid, and then it moves subsequently through the respective body portions of the nephridium. Okay, the fluid then gets alongside the ciliated narrow tube. Cilia basically can serve as an organ of locomotion. I'm sure you've heard of cilia as the organ of movement in the paramecium. Okay, but when you find cilia in the digestive system, for example, it aids in the movement or passage of materials across. So, by the way, it's important that you note that this coelomic fluid comprises of both useful substances like glucose and then unwanted substances like urea. So as they travel through, at this point now at the narrow tube, it moves across board. You find that the useful substances like glucose are reabsorbed back into blood circulation. Of course, you should also know that the nephridium is richly supplied with blood capillaries for the absorption of useful nutrients. And along the line, some nitrogenous compounds are actually also absorbed by some secretory cells before this fluid eventually gets to the muscular tube for temporary storage, as you can see over here. For temporary storage, as it moves across the middle tube, it gets to the muscular tube for temporary storage of materials, just like you have your stomach, okay, which holds digested food temporarily until it is transferred to the next point during the digestion process. A domestic fowl, for example, has a crop which serves a similar function, temporary storage of materials. Okay, here in the earthworm, we have the muscular tube which holds materials temporarily until eventually these materials get to the nephridiophore. The nephridiophore is basically the equivalent of the rectum or the anus in a man. Okay, at the nephridiophore, then the waste is discharged to the exterior of the environment. Okay, so this is quite an interesting process. Let's do a brief recap before we conclude. We started with an overview of the excretory system in earthworm, and we said the major excretory organ of an earthworm is the nephridium. Okay, and each segment in an earthworm except the first 3 segments and the last contains a separate nephridium. We learned that the nephridium has, each of these nephridiums have similar plans. They look the same basically, and then we said each nephridium has 5 portions: the nephrostome, the narrow tube, the middle tube, the wide tube, and then we have the muscular tube. All right, we moved on to discuss the content of the nephridium and see how substances move across the body cilium from the nephrostome to the ciliated narrow tube. It keeps being pushed along. Then the useful materials, glucose is absorbed along the line, and some nitrogenous compounds are absorbed by secretory cells until the waste products are now transported to the muscular tube for temporary storage, and then eventually expunged or expelled or given off from the nephridium to the exterior, the external environment of the organism. So next time you see a question asking you what part of the what organ of excretion is associated with an earthworm, you should be able to answer it very easily. Of course, the answer is the nephridium. Now let's get right into the next part of our teaching. All right, let's move on to discuss the excretory system in insects. Excretory system in what? Insects. The major excretory structure of an insect is the Malpighian tubule. The Malpighian tubule, of course, you can see it spelled out nicely on the board over here. We'll begin with a short overview of the Malpighian tubule and excretion generally in insects before we get into the mechanism of excretion in insects. Excretion is carried out in insects basically by the Malpighian tubule, of course. The Malpighian tubule is found at the junction between the midgut, where you have the ileum and the small intestine, and the hindgut, where you have the large intestine. The Malpighian tubule has a proximal and a distal end, of course, and the tubule, the internal cavity of the tubule has glandular epithelium, which comprises of microvilli, which serve the function of moving digestive materials around. Of course, just like you have the cilia, and when we talked about the previous discussion, we had the cilia there for the movement of material. Now we have the microvilli. All right, up next we'll look at the Malpighian tubule, by the way, before we get into more technicalities, is bathed in hemocoil or hemolymph. So, as man has blood, insects have hemolymph or hemocoil. It's important you bear that in mind as we discuss. Now on to the mechanism of excretion in insects. The microtubule, of course, is bathed by the hemocoil. The tissue, we are using cockroach, by the way, the cockroach as a case study as we discuss this. So the tissue of the cockroach produces some soluble nitrogenous waste products, which are basically potassium urates. The potassium urates is then expunged into the blood circulation, of course, and the distal end of the Malpighian tubule then absorbs this potassium urates. The potassium urate then combines basically with carbon dioxide and with water, forming 3 basic products. Number 1, potassium. Number 2, sodium bicarbonate, and then it proceeds to form uric acid. At the proximal end of the Malpighian tubule, the water and the sodium bicarbonate get reabsorbed. Okay, then we are left with crystalline uric acid, which is of course toxic to the body of the living organism. The crystalline uric acid then moves to what we call the hindgut. It's important we refer to the diagram. Actually, this is the gullet, this is the crop, the gizzard, the midgut, the ileum. We have the hindgut, the large intestine, where we are currently discussing, and as you can see, these spikes over here comprise of what we call the Malpighian tubules. Okay, so now the uric acid, which is a waste product, is accumulated in the hindgut, which is the large intestine, where it mixes with what? Feces, fecal matter. Okay, as it mixes with fecal matter, there is still some concentration of fluid here, so absorption of that fluid, actually reabsorption rather of that fluid, now reoccurs at the hindgut. Then afterwards, of course, this material, the waste material, then proceeds to the rectum and then expunged via the anus. So this is the summary of the excretory mechanism in an insect. It's important that you also note that Malpighian tubules occur in pairs of 6 actually. As you can see, we have my diagram is not quite complete, but we have a pair of Malpighian tubule, we have another pair, we have another pair, we have another pair, we have another pair, we have another one here. So 1, 2, 3, 4, 5, 6. But the actual number of the Malpighian tubules vary depending on the species of the insect you are dealing with. For example, in Blatia orientalis, we have in the male species we have roughly 148 Malpighian tubule and 186 Malpighian tubule in the female. So this is a really fascinating process. Over here you can see this is a section, a transverse section of the Malpighian tubule. You can see the nucleus, and you see in the internal cavity we have the microvilli comprising basically of glandular epithelium. So this is the summary of the excretory mechanism in an insect. Let's quickly move right to the next part of our teaching.
Yeah, hold up, wait. I'm about to cause an insurrection. I promise y'all I'm gonna be quick. Supersonic speed I'm running.